This series of videos is designed to teach people exactly how and why evolution works. While I'll be offering plenty of evidence for evolution, the goal of this series won't exactly be to convince people of evolution. This is because evolution is open to about as much reasonable doubt as whether or not the Earth goes around the Sun, and time is better spent educating people on the specifics of it. As far as the level of technicality that these videos will be discussing, they're pretty much suitable for everyone. I'll be discussing everything from the intro to DNA, intro to genetics, all the way up to epistatic interactions. So whether or not you're a biologist or you have absolutely no training whatsoever in biology, you'll still have a lot to learn, and hopefully the information will be presented in such a way that anyone can understand it and find it useful. I've been doing this long enough to realize that there are three types of people involved in the public debate. The first are people who are open-minded, intelligent, and understand the evidence for evolution and subsequently accept it. The second type of people are those who are open-minded, but they are not really sure of the evidence and they don't know what to think. They're fence-sitters, so to speak. However, if they understood the evidence, they would accept evolution, and they do have open minds. Thirdly, there are those types of people who, sadly, there is absolutely nothing whatsoever that you could ever show them. They will not accept evolution. Their minds are about as open and porous as a sheet of glass. There's really nothing whatsoever that you could do. As Ken Miller often points out, his colleagues will often come up to him and say, well, what if we just showed them this experiment, or what if we did this? Would they then accept evolution? And the answer is simply no. When these people view evolution as the way they do, and they're not willing to accept or process any data whatsoever, there's absolutely nothing that you can do to get through them. And as such, I'm assuming this attitude, and I'll be pursuing it, not addressing these people in my video. Quite simply, you can't fix stupid. This video, therefore, is aimed at the other remaining two people, those undecided and those decided. Now, those people who are undecided will get educated and be able to see the evidence so that they'll be able to better make an informed decision. Now, those who already have decided but don't really have extensive biological training will get that for free nonetheless. So, to begin, this first video is essentially going to be discussing exactly what evolution is, and more importantly, what it is not. The National Academy of Sciences recently called evolution the unifying theory of biology. This is because it unites virtually every independent discipline in biology so that scientific advances made in one can be universally applied to all others. Now, this has led to an explosion of research, and which is why evolution truly is at the core of biology. A famous biologist once said that absolutely nothing in biology makes sense except for in the light of evolution, and this is 100% correct. So, with that being said, I'll furthermore dive into exactly what evolution is and is not. To begin with, evolution does not state that human beings came from monkeys. It states that we share a common ancestor which is completely different. Second of all, evolution and God are perfectly compatible. Evolution says absolutely nothing about the existence of a deity whatsoever, and as this recent survey shows, 70% of scientists believe in a God. Furthermore, 99.98% of biologists accept evolution. So as you can see, there's clearly no discrepancy whatsoever. In fact, the past several popes have accepted evolution, and it's only in conflict with a literal interpretation of the Bible. Thirdly, evolution doesn't deal with how life arose on Earth. It only deals with life once it's already here. The same holds true for the Big Bang, many other theories which are commonly confused with evolution. The Big Bang and abiogenesis are theoretical physics and chemistry respectively. They have absolutely nothing to do whatsoever with biology and are about as related to evolution as they are the cell theory. Evolution is not something that one believes in, it's something that they accept. This is because evolution is a scientific fact. You don't believe in gravity, you don't believe in cells, you accept it and you acknowledge it. Now, evolution is not a belief also because it's testable, it's falsifiable, and it makes predictions. Hence, it requires absolutely no faith whatsoever, only acknowledgement. Next, evolution is not contradicted by really any evidence. Virtually every single ounce of evidence or observations that we can make about our natural world support evolution. Evolution was also not contrived to undermine any religion whatsoever. Basically, our awareness of it grew as we tried to make sense of the many observations of life that happened in a testable way. Evolution is not an accident or random process. There are some random components of it, however the vast majority of it is due to natural selection, which is by definition a non-random process. Evolution is also not something which happens to specific individuals. Instead, it happens to populations. Evolution is not something that only happened in the past. It's a current process, it has happened, it's happening now, and it will happen in the future. A quick comment before I progress too much. If in this video you find something that you want to learn more about, don't hesitate whatsoever to make a comment. I'm pretty good about responding. Furthermore, you should also take a look at the berkeley.edu website. They've got a fantastic evolution page, and it'll explain and reinforce a lot more of the concepts that I'm explaining to you guys. Also, another link that I would highly recommend you guys is this Talk Origins FAQ. This essentially rebuts virtually any creationist argument that you could ever dream of. If there's something so obscure that they're very good at pointing out randomly, you can find it here typically. And it's a wonderful resource, and highly encourage you guys to take a look at it whenever you run into any issues whatsoever if you don't have much scientific training, or if you're simply interested in browsing. Your time will be well spent. In a biological sense, evolution is best described as descent with modification. This leads to changes in gene frequency within a population over time. The idea is that an environment selects for an organism that's best adapted for survival in it, and they're encouraged to breed. This concept leads to a change in gene frequency over time, as well-adapted organisms breed at an accelerated rate, and their genes constitute a larger portion of the gene pool. As time progresses, populations become isolated, subdivided, and undergo different changes at different rates as they become very well suited to their environments. Now, after two populations will accrue enough differences, they'll no longer mate. When this happens, a new species is formed and they're said to have speciated. Now, this happens more common and rapidly than most people think. For example, scientists have observed 40 speciation events in the last 15 years alone. This is directly influenced by many factors, including generation time, which I'll get into in later videos. However, the point being that it's very common. Actually, some interesting news about just this topic came out last week. In 1971, 5 adult pairs of Italian wall lizards were transplanted to the island of Croatia. Now, they were an invasive species and essentially overran the indigenous population there. Go scientists. Well, the Croatian War of Independence broke out in 1971, or shortly thereafter rather, and nobody was allowed on the island, at least not scientists, to study the effects until recently in 2004. What they discovered was that not only had the new lizards overrun the island, but they had completely evolved. And not only had they evolved, but they had evolved at a rate that even surpasses what evolution would predict. In other words, this is a phenomenal example of lizards evolving at a rate that's much, much higher than one would predict. In fact, these lizards the lizards that were found developed cecal valves, which are muscles between the large and the small intestine that slowed down food digestion in fermenting chambers, which allowed their bodies to process the vegetation cellulose into volatile fatty acids. So essentially, they evolved an expanded gut to allow them to process these leaves. This is adding a brand new structure in just 30 years. This is a wonderful example of a new species forming and the addition of a brand new structure, which is not originally present in the organism's genome, because keep in mind, we have the original population of lizards as well back in Italy. So suffice it to say, we actually know a lot about this. It's happening continually, and it's an ongoing process that we learn more and more about every day and every year. This video has been a pretty basic introduction into exactly what evolution is. Now, my next videos will be focusing more on the biology of evolution in general, and specifically the next one will be focusing on the 4 forces which drive evolution. So stay tuned, guys, and once again, thank you very much for your time. As I stated before in my prior video, there are 4 main forces which drive evolution. They are natural selection, mutation, genetic drift, and gene flow. Now, I'll go into all 4 of those at length. So to begin with, we have mutation. Mutations are random changes in an organism's DNA. Now, mutations can occur in several places. For example, it can occur in a somatic cell, which is a cell of your body. For example, you can have one of the skin cells on your arm undergo a mutation. That'll be influenced by you, and it won't have any effect whatsoever on your offspring. However, it can also occur in some of your gametic cells, such as your egg or your sperm. If you have a mutation in a gamete, and that egg or that sperm goes on to produce a child, that mutation will go on and be present in all of your children's body cells. Mutations can typically have one of 3 effects. Some of them are bad. They decrease the overall fitness or survivability of the organism and are not selected for. Now, the second type, the vast majority of them are neutral, meaning that they provide no change whatsoever to the organism's fitness, and it is neither helped nor harmed by the mutation. The third type, which is the rarest, are beneficial mutations. These help the organism by increasing its fitness and the likelihood that it'll reproduce and pass on that mutation. Now, specifically, there are several types of mutations in the sense of inversions, deletions, additions, insertions, and things like that. However, I'm going to be discussing that in my next video, which will essentially also give you guys a brief introduction of DNA. But until you guys understand what DNA is and how it works, me going into the nitty-gritty specifics of mutations really won't help you. So I'll leave it as is at this point. Nevertheless, mutations are random. A very important thing that people don't seem to understand is that an organism does not know it has to mutate. For example, let's say that there are a group of green beetles and there are some mutated brown beetles. The green beetles are the natural wild type, but due to a change in the pigment gene, it'll also produce brown. Now, let's say that there's a bird that can only see and is only used to eating the green beetles. It won't touch the brown beetles. Now, did the green beetle, did it know that it had to mutate and become brown so that the bird won't eat it? No, what happened is that it passively mutated and the environment selected for those which were brown and made those which were brown more likely to pass on their brown genes. The second force driving evolution is natural selection. Natural selection is, by definition, a non-random process. Another common mistake is that people seem to think that the organism does the selecting or it's selected for one way or another, but it is actually the environment which is doing the selecting. As I said before, mutations provide the raw material, the raw genetic material for natural selection to work on. You can kind of think of it as mutations creating a static image and natural selection being the refinement of that image to produce a picture. Actually, take a look at this image that I'm showing right now. It's a computer model made by CDK007. I highly suggest you guys check out his vids, which start off by with a random static field, and by selecting only the ones which are likely to improve and closely match the picture that's desired, we end up with an overall South Park picture, or a picture of a South Park character as the net result. Although I absolutely loathe using the term survival of the fittest, this is essentially what it's referring to. In a nutshell, an organism which has genes which allow it to be very well adapted to its environment is more likely to be able to survive and pass on those genes. Over time, those genes will represent a relatively larger percentage of the gene pool of that population. The third force driving evolution is genetic drift, which is a random process. In all actuality, it probably is understated in the sense of genetic drift probably has a bigger effect on population genetics than most people realize, but nevertheless, it is a random process, and it's often overlooked, but it's very important. Now, evolution works best in very, very small populations, because in those populations, there's a lot less averaging, and big changes can accrue very, very quickly. Kind of think about it with a law of averages, and if you've ever done any statistical work, think of outliers. For example, let me say that I'm taking the average weight of a baby, and I have 5 babies. 4 of them weigh 15 pounds, and 1 of them weighs 3 pounds. Now, this is completely hypothetical, but think about the effect of that 3-pound baby on the average weight of all those 5 babies. Now, on the other hand, let's say that I take the birth weight of 100 babies, and 99 of them weigh the 15 pounds, and there's still that 1 3-pounder. So if you have the large sample size of 100, the average birth weight will be much closer to the reality of 15 than it will in that 5, or in the group of 5 individuals. Now, the 3-pound baby is termed an outlier because it greatly skews the statistical results that you're looking for. It's just a random, aberrant data point, which can basically screw up on trends. Now, for evolution's sake, evolution works best, as I said before, in small groups. Now, this is because those random genes can be compared to outliers. Any new mutations that would be great to be selected for are often drowned out if you have an extremely large population. Whereas in a small population, those random genes will contribute much more, and they'll be much more powerful and pronounced. Now, this is where genetic drift comes into play. Genetic drift is a random change in gene frequency over time, which is due purely to chance. Now, contrast this with natural selection, which is actually not due to chance, which is, the environment is selecting more fit ones. In this one, this actually is an accident. If you have a population of 5 beetles and half of them are brown, half of them are green, and you just step on some, and you just by chance knock out all the brown ones, well, then you're going to have a population with only green, and the brown's going to be eliminated from the population. This is gene flow. And what this can do is when a population or a large population splits into two small ones, it can create large differences between those two populations over time, and that is exactly how those two populations will split, and evolution and speciation stems from that. So a large population with tons of mixing of different genes from different individuals makes it hard for any differences to accrue. Large populations tend to be relatively homogeneous, and although there are a decent number of differences among individuals, everything gets sort of blended together. And this works against speciation because speciation relies on differences within individuals and differences within populations. In other words, if you have a big population that's subset into two smaller ones, that's exactly how speciation occurs, because genes are not mixing. So mixing of genes tends to slow down evolution, and that's actually what the fourth concept is, and that's gene flow. So with regard to gene flow, let's go back to the drawing board and let's take a big population and actually examine how speciation happens. Note that I'll be covering this very much more in depth in later videos, so if you don't understand it now, I'll cover speciation much more. I'm not planning on doing much of it here. However, speciation typically happens, there are several types, but it's when you have a large population and then a small population buds off, or there's some sort of barrier between the population. It can either get segmented in half or whatnot, or anything like that, basically to prevent the genes from being exchanged from the two. Now, gene flow is exactly that. It's genes being exchanged between the two populations. So you can think about it as a smaller subpopulation is trying to accrue a lot of genetic differences from the main population. However, because there's still mating going on, it prevents and it slows down those differences from accruing, because the genes keep on getting mixed together and the main population genes keep on getting inserted into the small founder genes. An easy way to remember the differences between genetic drift and gene flow is you can just think of genetic drift as the genes in the population just kind of randomly drifting away and being removed from the population. Whereas with gene flow, you can think about genes flowing between the two populations, because that's exactly what it does. I'll end this video here. My next video will be a basic introduction into DNA, and it'll also be explaining the different types of mutations. Oh, lastly, a couple of you guys have asked me about a lot of the images and the demotivational posters and whatnot that I use in my videos, so I've decided to create a photobucket album and upload them to there. It's photobucket.com, username is Don Exodus, but I'll go ahead and include the link to the album in the video description. So I created the vast majority of them. Feel free to use and distribute them however you guys. The third video in this series will be designed to help you guys understand DNA. Now, there's a lot of mysticism surrounding DNA in general, so hopefully this will help clear it up for you guys. There's going to be a lot of information packaged into this video, so if at first you don't understand it, by all means watch the video again, because it is relatively important information, and in the next video, the fourth one on mutations, you're going to need a lot of this information to understand exactly the different types of mutations and what their implications are. This is the structure of DNA from a little bit of a distance. I'm sure most of you guys have seen this. It's the classical double helix structure developed by Watson and Crick many years ago. Now, there are essentially three parts to DNA, and that is there's a phosphate side rails and sugar. Together these are termed the phosphate-sugar backbone, and they're common throughout DNA. They're not what changes, and they don't code any information really. Now, what does code the information are the rungs of the ladder. Those are called nitrogenous bases, and there's A, T, G, and C that's found in DNA. These are the subtle differences between the A's, the T's, and the G's and the C's. As subtle as it looks, however, these make all the difference in the world, and they're responsible for coding every gene in our body. Now, it's important that you guys understand also that all DNA does, by and large, is encode proteins. That's it. And so I'm going to be covering proteins a little bit later on in the video. Also note that what you guys know as chromosomes are really just DNA from a distance. See, DNA wraps around itself and wraps around these things called histones and solenoids and just gets all sorts of balled up and tangled up. And when you tangle up enough of a mass of it, you can actually see it, and that's what chromosomes are. But nonetheless, the information in DNA can be read in the order of the nitrogenous bases, going from a 5' end to a 3' end. It's not important that you know the distinction, but simply know that there is directionality to it. So you can start by reading the genetic code, as I stated before, by the order of the nitrogenous bases, that is the A's, the T's, the G's, and the C's. Now, every 3 nitrogenous bases are collectively known as a codon. So one codon is equal to like a CAG or a GAC or something like that. And each codon encodes an amino acid. Now, multiple amino acids strung together encode a protein. So think of it as DNA, every 3 bases equals 1 amino acid, many amino acids equal a protein. And this is very, very simple and fundamental to biology. Now, there's also what's known because of this as a reading frame, which means that if you were to screw up DNA by what's called a frame shift mutation by either adding a base or deleting a base, everything downstream from that would be garbled. Because keep in mind, every 3 bases equal a codon, so if you're taking one of the things out of it and shifting everything over, you're changing the order of where the codons stop and begin, and they're going to consequently encode entirely different amino acids and entirely different proteins as well. So this is very important, and the reading frame must be maintained in order to translate the proper amino acid that's desired for the given protein. It's also important to understand that genes have structures as well, in the sense of part of a gene is what's called the structural domain, and that encodes for actual proteins. That's the functional product. Whereas the other section of a gene is called the regulatory section, and that acts as a switch. So it's not either you have a gene or you don't have a gene. More often than not, genes can be easily controlled simply by switching them off, as opposed to completely deleting them. This is oftentimes what happens in evolution, in the sense of a gene will just get switched off, but the structural part will remain. Now, also keep in mind this is how atavisms work, which I discussed in my Proof of Evolution 3 video, where the genes don't simply get deleted whatsoever, they just get turned off, and in an atavism, they just get reactivated or switched back on. So it's very important also to understand that this is a key mechanism that can be used to regulate genes also. Okay, so you've got your DNA, you've got your GAC, TAG, you've got your reading frame, you've got your codons and all of that, but how do you actually make a protein out of it? Well, what has to happen first, and this is what's called the central dogma of biology, is that DNA has to be transcribed into RNA. Now, the difference between RNA is very simple. It's simply that DNA is lacking in oxygen on one of the carbons. That's the only difference. That in DNA is double-stranded and RNA is single-stranded typically. So what happens is there's an enzyme called RNA polymerase which goes along DNA when it's time to be transcribed, and it creates an RNA copy of the gene. Now this RNA copy then goes through what's called translation, and it meets a ribosome, which is an organelle in the cytoplasm of a typical cell. So the RNA meets up with the ribosome. It then goes and through various other types of RNA, such as tRNA and whatnot. The mRNA message and the codons contained in the RNA are translated into amino acids. The protein is assembled by the ribosome and it exits. So what you have here is a message going from DNA to RNA to protein. And that is a central dogma of biology, and it's very important, and that's what controls absolutely everything in our body. One more interesting thing to note before we move on is that there are a lot more combinations of A, T, C, and G than there are possible amino acids. As such, several codons will code for a single amino acid. For example, CCT, CCC, and CCA, and CCG all code for the same amino acid. This is what's called as the redundancy of the genetic code. And it's also very important because oftentimes what happens is it's just the very last letter that's changed. Because keep in mind, CCT, CCC, CCA, CCG, it's only the last letter which is changing, and as such it's all the same answer. So pretty much anything with CC blank is going to be making a proline. And that's very important, and it's the foundation of what's called the wobble hypothesis, which I really don't have time to get into here, but if you want a good read, Wikipedia it. Another very important concept that you guys understand is that proteins, their function is absolutely critical and dictated by their structure. Proteins work typically by what's called like a lock and key model, and that is that they will only act with their receptor, with their substrate, with whatever, if the structure is absolutely lined up. Because keep in mind, DNA is coding for 3D molecules in space, and the structure of which is extremely important to their function. So proteins serve many functions. They serve as enzymes. They're the foundation of muscle. They serve as structural purposes too. They're collagen. They also serve as transport. They serve as antibodies. They're the workhorse of the human body, and any body really. And they're fantastically important. And again, they're all encoded by DNA. And once again, remember, the structure of the protein is what determines its function quite often. So how is the structure determined? Well, there are 4 substructures. There's the primary structure, which is the amino acid sequence, that is the order of the codons, the A, T, C, G, A, T. Those are what's called the primary structure. It's just flat on paper. That's what you think of. Then there's a secondary structure, which is alpha helices and beta sheets, and that's not really important. So when you form all those together, then you get the tertiary structure, which is a folded peptide being protein. And then when you get several peptides interacting with one another, that's when you get to the quaternary structure, and then you have like a fully formed and functional protein. So what determines then exactly how the protein will fold? Well, it's actually intrinsic, given by the primary structure that is the amino acid sequence. See, amino acids have different physical properties in the sense of some of them are basic, some of them are acidic, some of them are polar, some are nonpolar, some of them have sulfur, and it all corresponds there. Whenever you take a protein and you put the amino acids together in the proper way, given that the conditions are right and it won't be denatured, it'll automatically fold into the correct position. So from the primary sequence, that is the DNA sequence, and the primary structure of the protein, which is the A, T, C, G, etc. of the amino acids, you get how the protein will fold, and it'll fold the same way every time. So remember, the protein, the way that it folds, is what determines its structure. So I hope that this is all coming together for you guys, because remember, you had the DNA, which goes to RNA, which goes to amino acids, which goes to protein. And the DNA is encoding which amino acids are going to be made, and those amino acids have certain properties which dictate how the protein will be folded. And as such, because those amino acids have those properties and are dictating how the protein's folded, that'll determine the protein function. So I know this is very complicated, and I know I'm doing a lot of redundancy, which has to be annoying for you biology majors. However, it's a complicated concept, and redundancy certainly won't hurt in helping the newbies get it. So make sure that you understand this concept because it's very important. So this will essentially conclude my introduction to genetics, and this is very important to understand because my next video is going to be on mutations and the different kind of mutations, and when you understand exactly how DNA works, then you can understand the implications for each mutation. So thanks again for your time, guys. So those of you guys who are still kind of curious about it, let me go through it one more time real quick. DNA has A, T, C, G, etc. Those are nitrogenous bases. Every three of them make a codon. They code for one amino acid. Each amino acid has various physical properties which encode and determine the way that the protein, which the amino acid makes, folds. So again, DNA goes to RNA, hits a ribosome, turns into amino acids, and which fold correctly. This video is going to be on mutations, and it's also going to represent the point in the series in which we'll be moving a little bit away from standard general biology and actually into biology as it pertains to evolution. So, as far as mutations go, there are several types, and there's a lot of mystery surrounding mutations as far as the general public is concerned. There are a lot of misconceptions in the sense of that it'll make this horrible creature all the time, that they're always bad and things like that. Those simply aren't the case. What mutations are just changes in the DNA that often arise from copying errors whenever DNA is being transcribed. You can just think about it as if you were to transcribe or write a book by hand, just make a copy of it. Instead of writing the cat ate the dog, you wrote the bat ate the dog. That would be an example of a mutation. Now, there are several types of mutations, and I'll start to go into those one by one here in a minute. Also note that this video is going to be covering the common misconception of mutations can't add any new genetic information, as I'll show exactly how that can happen and examples as far as where it has happened. An important thing to understand is that mutations can occur anywhere along the DNA strand. It can happen in the structural portion of a gene, making a completely different three-dimensional protein, and thus having a different function or be functionless. It can also happen in the operator portion of a gene, rendering it always on, only on in the presence of a certain molecule, or always off. It can also occur in non-coding regions. That is, there are certain segments of DNA which don't code for any protein, don't code for any gene, and a mutation can happen there with, relatively little consequence whatsoever. It's also important to note that an organism doesn't know that it needs to mutate. It's not anything that you can control, it simply happens. Also, as much of it happens during the copying of DNA, there are many proofreading mechanisms that DNA goes through in the sense of after it copies DNA, or after a cell copies DNA when it's beginning to replicate, it'll go through various mismatch repair mechanisms and things like that to ensure that the new copy is correct. And while these safeguards against mutation correct the vast majority of them, every now and then mutations do get through and passed on. It's inevitable. Each of us have plenty of them in our bodies going on every day and every hour. Nevertheless, these mismatch repair mechanisms correct the vast majority of mutations, but they do occur at a set rate, in the sense of every so many generations you will have X mutations passed on to your offspring, or every X cell divisions you will have X number of mutations. Furthermore, we can also take a look at two organisms, related, unrelated, same species or not, and compare the number of mutations that they have. And since they occur at a set rate, we can predict, based on the number of mutations between them, the last common ancestor. This is commonly used, and it's really not up for debate. So, there are also, as I mentioned before, several types of mutations, and I'll start going into those now. A type of mutation called a substitution can occur if you substitute one nitrogenous base for another. So for example, in the sentence, the cat ate the dog, if you change that to the bat ate the dog, changing the C to the B would be an example of a substitution. Now, this doesn't always lead to a different protein being made, because if you recall the wobble hypothesis, which I showed by this picture earlier in my third video regarding DNA, if you remember, the last codon, or the last base in a codon, can oftentimes be coded for the same amino acid. In other words, you can have AAT and AAC both coding for the same amino acid. So when this happens, there really is no change because you're making the same protein. However, if it does substitute for something else, you can get a completely different amino acid, which can affect the folding and oftentimes structure and function of the finished protein. So another type of mutation can be called just a deletion. It's where you simply have a gene and just a piece of DNA gets lost. It happens most of the time. This is deleterious, however, not always. It just depends on the segment of DNA. For example, if this happens in a non-coding region, it typically won't really hurt anything. Another type of mutation is called an insertion or deletion, and these are called frameshift mutations collectively. If you recall from the third video, DNA has a very distinct reading frame, and that every 3 codons will equal 1 distinct amino acid. And it goes based on every 3. If you were to insert something or delete a specific base, you would shift the reading frame completely. This results in virtually every amino acid that's coded after that to be completely changed to something completely different. And as such, this will affect the protein folding and the structure and the function. Now, most of the time this induces what's called a stopped codon, and it causes the transcription to stop, so you'll get a short, non-functional protein, but every now and then, in the case of myelinase, you'll get a completely different protein. That is, if the stop codon isn't encountered for many bases along the way. But nevertheless, insertion and deletion mutations are extremely important, and as they allow for the creation of novel enzymes, very rarely, but it absolutely does happen. So if it happens with one, you'll have typically just completely different amino acids following. If it happens in two places, that is, if you have it on position 1 and 2, you'll also get completely different amino acids. But if you have it in 3 times, that is, you have 3 insertions or deletions, that one particular amino acid will be messed up, but everything afterwards will be the same. So think about why this is, because if you remember, it's a 3-base reading frame, so you'll just knock out one portion of the amino acids, and that's very important to understand. The final type of mutation that I'll be going into is called gene duplication. This very commonly happens in that you have a gene and you essentially just duplicate it so that you have an extra copy. So keep in mind, if this gene were to be making a very essential protein for the cell that it needs to live, if it were to be deleted, the cell would die. However, if you were to have an extra copy of it, and something were to happen to one of those extra copies, well, the cell would be fine because it would still be making the original one. And this is what often happens, or it would still have the original intact gene and it'd still be making the original functional protein. So if you go back and actually think about what this means, that means that you have an extra template. You've got this duplicated gene, you've got your other one functioning fine, that you can have all sorts of mutations occurring on in, and basically with no ill effect. This leads to basically kind of your cell can experiment, although it doesn't actually know that that's what it's doing, but it can accrue large differences, all sorts of mutations, and make all sorts of new and interesting proteins, most of which have absolutely no function whatsoever, but occasionally some of them do. And this is exactly how new genetic information is added, by gene duplication followed by subsequent mutation. And myelinase is an excellent example of this. So to tie a lot of these concepts together, myelinase, which is a novel enzyme, a brand new enzyme that's was found in bacteria, enabling them to digest nylon. Now we know that this wasn't present in the environment before because nylon has only been around for a couple decades, and since it wouldn't be having any other function, which the nylonase enzyme doesn't because it's very specific for its substrate, it would have accrued lots of mutations had it been there from the beginning, as there's nothing selecting against them, and remember, mutations occur at a set rate. So we've got this new enzyme. What happens, or what led to its formation, is there was a gene that was duplicated, and from that a frameshift mutation occurred, leading to all sorts of completely different amino acids being formed just from this one mutation. And keep in mind that although the odds of this happening are extremely small, when you have billions of organisms on Earth with very small generation times consistently multiplying over millions of years and billions of years, I mean, this is exactly what happens. These things accumulate, and they're responsible for the amount of diversity that we see today. So again, quick recap. Nylonase, a brand new enzyme that was formed from the insertion of one amino acid that caused a stop codon 392 positions away. Now, what this did was it allowed for the encoding of all new, completely different, completely random amino acids, which just happened to change the way that the protein would fold. By changing the folding, you're changing its structure. By changing its structure, you're changing its function. So it went from a protein that could not digest enzyme or could not digest nylon to one that completely could, which is exactly how new information is added. And this has always really puzzled me, this argument of no genetic information has ever been added, because it's seen every day. It's nothing new. I mean, we've known about this for years. It's basically in any high school level biology book. I mean, it's fact, it's not up for discussion, and it's always puzzled me exactly why this argument has persisted. I guess, like all things with creationism, it really just kind of stems from misunderstanding. Another example of how you can add new information is basically the addition of any trait. If I were to have any mutation, for example, if I were to have brown hair and I would have a mutation there, the population of humans would have a mutation that would cause you to have blonde hair, that'd be new information. Even if you are deleting part of the gene that codes for the pigment of hair to one that makes brown pigment to one that makes no pigment or blonde pigment, even if it were a loss of information, it would still be a gain in information because you have an additional type of organism that can be formed. You can either have a brown-haired one or a blonde-haired one, whereas before you could only have brown-haired. This is new information as it's a new trait. Whether or not it comes from the net loss of information or a net gain, it's a complete misunderstanding of how DNA and genetics work in general. This has been a pretty brief introduction to mutations and exactly how they work and what kinds there are. There are several kinds that I know that I didn't list, but they're frankly not relevant to this discussion. Check out the link in the video description as it's to a Wikipedia page regarding mutations, and it's a fairly decent read. There appears to be a lot of ambiguity in the public regarding natural selection and exactly what it means. I think that Charles Darwin summed it up pretty well when he said that it's not the strongest of the species that survives, nor the most intelligent, but rather the one that's most responsive to change. And that's something that's extremely important to understand about natural selection, and that it's not survival of the fittest by any means. It's merely that in a changing environment when speciation happens, those which are able to adapt to those changes are most likely to survive and pass on their genes. There are several things, or components rather, which actually make up natural selection and lead to it. First, there has to be variation within the population for natural selection to act on. Second of all, there has to be differential reproduction in the sense of organisms with certain traits are more likely to reproduce and pass on their genes than others. And thirdly, there has to be heredity in the sense of the genes that are actually being selected for actually have to be genes and be able to be passed on. So the combination of there being different genes, some genes being more likely to be passed on than others, and them being genes which are able to be passed on, period, are all a component of natural selection. It's also important to realize that evolution is completely dependent on the environment. For example, a bacteria that produces enzymes to degrade glucose constantly is fantastic if they're in a glucose-rich environment, but if there's no glucose whatsoever in the environment, that bacteria is actually going to be at a disadvantage. Why? Because they're spending all this energy making this enzyme to convert something that's not there. So it's important to keep in mind that something that's advantageous in one environment will actually be a disadvantage in another environment. So within evolution, it's the environment that's actually doing the selecting. It's merely a statistical event where certain organisms with certain genes are more likely to pass on those things and survive. Now, in this sense, it's also important to understand that evolution or natural selection doesn't need to do anything. It doesn't want to do anything. It doesn't try to do anything because it's not a conscious entity. It merely happens. So whenever you say that this organism needs to or this organism wants to, that's not what happens. Indeed, it's just the environment is selecting organisms which possess a certain trait, making them more likely to pass on that trait. So always be extremely careful of referring to natural selection when using verbs like those, because it's not the case. So now let's use an example to clarify exactly what's meant by natural selection. In a lot of my prior videos, I've used the example of a population of green beetles upon which there was a mutation which caused the introduction of a new color beetle, a brown beetle. So let's say that there's a bird which only eats green beetles. It absolutely can't stand the brown ones, so it won't eat them. What'll happen eventually is that the beetles, or the green beetles rather, will be picked off at a disproportionate rate. They'll be less likely to reproduce, and the brown ones, consequently, will be more likely to reproduce and pass on their genes. So what this will do is it'll lead to a difference in gene frequency over time, because before, the population was mostly green. However, now, over time, because all the green ones are being killed before they can mate and pass on their green genes, the population is going to become largely brown. And this differential reproduction is exactly what is important, and the cause and the main driving force for the distribution that results from natural selection. Now, it's also important to recognize that natural selection isn't making any new traits. That's done by mutation, and natural selection only deals with what's already there. In my next video, I'll be discussing some of the constraints of evolution, and specifically the constraints of natural selection, and it'll kind of go hand in hand with this video. And you need to really understand both to get a good feel for natural selection and exactly how it works. Now, another often overlooked task that natural selection does is it prevents DNA from being mutated into nothingness. We know that certain organisms undergo mutations at a certain set rate. For example, let's say that there are 2 organisms that undergo 10 mutations every generation. There are 1,000 mutational differences between them. Given the number of mutations between them and the mutation rate, we can tell that there are 100 generations apart. This is important because over time, these mutations will accumulate in genes, and the genes will become functionless and won't resemble anything of their ancestors, and they simply won't work. So what's to prevent all these mutations from accumulating in a negative way? And the answer is natural selection. When an organism has a bad mutation in an essential gene, what will happen is natural selection will prevent it from breeding, and as such, that negative mutation won't be passed on. Also, organisms with good mutations will breed at a higher rate, so it has a higher chance of passing them on. An example beautifully illustrating this quality of natural selection can be found in nylonase. Some people could simply argue, well, how do you know that these bacteria weren't created with the ability to digest nylon? And the answer to that is nylon's only been around for 70 years. So until that time, there is no selective pressure to weigh out any bad mutations. And because, keep in mind, mutations that are occurring at a very distinct rate, mutations would have inevitably happened in the nylonase gene, rendering it ineffective. Because, keep in mind, there's no nylon around, so those bacteria that can't digest it aren't going to be at any disadvantage to those who can. That disadvantage only arises that advantage only arises once nylon is developed by humans in the past 70 years. So understand that with time, all genes will fill up with mutations. It's up to natural selection, which is a non-random process, to take care of those. For further information, if you still don't quite understand this concept, check out the CDK video that I'm attaching. Lastly, I wanted to touch one last time on that quote with Darwin that I started off with. That is, it's not the strongest or fiercest of the species that survives, instead it's the most adaptive and responsive to change. Let's go back to the beetle example. Let's say that there hadn't been a lot of genetic variation, and the beetles wouldn't have been able to respond to the prey, or to the new bird, and there was just green beetles. It's very likely that they could have simply been wiped out. The beetles could have gotten bigger, but that wouldn't have really done anything in the face of the bird. It would have just given them a more nutritious snack. Instead, what the ones that survive, and the species that survive as a whole, are the ones with the greatest genetic variation that are able to overcome these obstacles as they arise. This could also be seen with what's called a predator search image, and that is, over time, the percentage of the green beetles would decrease, so the birds then would shift their focus onto the brown ones. They would change their predator search image, and instead of looking for green ones, they would look for brown ones. Again, if the beetles didn't have any green ones left, or they were just only brown to begin with, they didn't have that genetic variation, they would also, again, be easy for them to be wiped out. So the adaptability and the responsiveness to change is a very important element of natural selection. It's not just about being big or bad or strong or survival of the fittest. It's about being the most responsive and adaptive. Because environments aren't static.
they change, and with what's good and well suited in one environment will be poorly suited in another. So it's up to the organism to be able to change, and those that can change with the flowing environments and be recently or decently well adapted in both of them, those are the organisms that'll survive. Lastly, I wanted to take the opportunity to thank once again everybody who supported me. I actually sent all of you thank you responses. However, many of them came back as undeliverable, so even if you didn't get one, understand that I did try and I am genuinely grateful. But I'm genuinely grateful to anybody who wants to learn or has anything that I have to say, or listen to anything that I have to say, period. So again, I can't thank you guys enough. I actually just got my new mic yesterday. I'm talking on it now. Still tinkering with the software a little bit, but I think that the audio quality is definitely improved, and it's just got nowhere to go but up from here. Also, news-wise, I think there's going to be an interesting event going on with a couple of the head honchos regarding evolution on YouTube, that is, Thunderfoot and company. So we might have a little surprise development coming up for you guys in the near future. So once again, thank you, and I hope this helps, and I'll be happy to answer any questions. Have a good day, guys. Evolution has many constraints. Upon looking at the natural world and examining the way things are organized, we see that many of these systems are designed with the evolutionary constraints in mind and develop only according to evolutionary constraints. And this itself is a pretty powerful piece of evidence for evolution. It's best to think of evolution as a sculptor, not really as an engineer. Although there are dozens of examples of evolution creating new structures, it does best simply modifying what's already there. A wonderful example for this are camel toes, and I'm not kidding. As you know, camels have fleshy pads. They've got 2 toes because they've lost the other ones. They share a common ancestor with all, tetrapods, so they originally had 5 digits, but they lost them, and now they just remain with 2 fleshy pads. Now, why would this be? Well, you need to understand the history of the desert, then that's where the camel lives. The desert wasn't always as such. The camel originally lost the toes to make more of a hoof-type structure, which would be excellent for running on fertile ground, which is exactly what deserts used to be before they dried up. But since the desert's dried up, then what exactly is the camel going to do? Well, it could regrow those lost toes, but again, that's difficult, and because they've been not being expressed for thousands of years, the genes would likely be subject to mutation because natural selection wouldn't be correcting them. For more information on this, see the last video, which was on natural selection. So regrowing them is an unlikely option. Instead, it's best suited to go ahead and modify those 2 toes into one that's good from running in fertile ground, like a hoof, to one that'd be great to running on sand, somewhat like a snowshoe. So how this would happen is it would take the 2 remaining toes that it would have, and it would flatten them out and make fleshy pads in between, and support webbing, which is exactly what the camel has done to make its toes ideal for running on sand, as opposed to on fertile ground like deers and horses. Another fantastic example of an organism evolving under evolutionary constraints can be the supracoracoideus tendon found in birds, which is responsible for the upstroke of the wings. Now, think about it from an evolutionary perspective. This organism has to evolve something which will allow the wing to upstroke, to get back, and so it's able to repeat the power stroke and fly. Now, there are 2 ways that it could do this. One is that if you were to create a brand new tendon, it would have to go from basically the back, which is where you'd put the muscle, which would be used to elevate the upstroke of the wing, onto the wing, and it would just be a straight shot. The other thing, however, and why that's not really feasible for evolution, is because the muscles which are responsible for the upstroke are located on the chest. So instead of being designed from scratch, what the bird has to do is kind of rig what's already there and make its supracoracoideus tendon insert from the wing all the way, going up around the back, over the arm, and back and inserting into the chest muscle where it originates. And once again, this is completely not incompetent, but it's not the best way to do things. But that being said, it's the only way that it could have evolved. And, of course, it's exactly what we see. So, being able to understand the constraints of evolution and knowing what to look for can be a powerful piece of evidence for evolution as well, in addition to simply understanding it. Another thing to keep in mind when examining how a trait might have evolved is that there are trade-offs for virtually everything. Let's take a cat, for example. Now, cats need to be able to do two things, which are run fast and grasp prey. Well, unfortunately, the structures which would make each of those ideal are oppositely suited. For example, to run fast, you would want smaller surface area limbs, for example, like a horse's limb, so you wouldn't want to have these big fluffy paws. However, for grasping prey, you can't really grasp prey very well with a hoof, you need claws. So, for a cat to be able to do both things, it has to compromise and meet in the middle, and this is important also. Another thing to understand is that there are embryological constraints for evolution too. For example, just take a look at the recurrent laryngeal nerve. Now this is a nerve that comes off of the brainstem from the vagus nerve. It loops around, goes all the way down from the brain, loops around the arch of the aorta by the heart in the chest cavity, then goes all the way back up to the larynx, which is a several-foot detour. And why does this happen? Well, of course, the much more natural or the obvious thing to do would just have it go from the brainstem to the larynx, but because it develops a certain way, it can't do that. Because this nerve originates from the sixth pharyngeal arch, and yes, those are those dreaded gill arches, it migrates. Structures have to migrate out of the head into certain places of the body, and due to the differential migration of different things in different arches, it's stuck this way. So, although it would be ideal to have it simply run from the base of the brain into the larynx, it can't do that. It develops a certain way due to its evolutionary history, and it has to adhere to that. Another oddity about evolution is that when a trait is no longer needed, instead of simply scrapping it or throwing it out, evolution finds an alternate use for it. A perfect example of this is Meckel's cartilage, which started out as a lower main element in primitive first gill arches. But as life evolved, the structure was really no longer needed, so does evolution throw it out? No, instead it finds an alternate use for it. In human beings, instead of supporting gills, it just becomes this phenomandibular ligament. And this is just one more example, Meckel's cartilage, like virtually every other of the 1,000 components of the human body, you can see them and trace their homologs all throughout phylogenetic trees and throughout evolutionary history. So the evolutionary history of Meckel's cartilage is also extremely interesting, and I'm putting a link to it right here. So definitely be sure to check that out if you have some spare time. The remainder part of this video is going to be relatively new information, and many people typically don't understand this. However, it's a very important aspect of evolution, and it's the answer to when somebody asks you, well, why didn't X evolve this way or that way? It would have been much better. Epistasis and pleiotropy, which are two new concepts, are typically the answer as to why that didn't happen. So to begin with, epistasis is literally one gene being acted on by other genes. It means stepping on. So you've got many genes controlling one trait. For example, if I've got a gene for really tan skin, that's fantastic, but if I've also got a gene making me an albino, it's not going to matter. And there are many other examples of this, but that's just one. Even more important is pleiotropy, which is where one gene is controlling many traits. And a wonderful example of this could be Tyrannosaurus rex arms. So let's say that, you're a dinosaur. Tyrannosaurus rex, any dinosaur with very large head size, or jaw size specifically, would be very well selected for. It could eat more things. But unfortunately for that, the gene which is controlling jaw size is also controlling arm size in an inverse proportion. So to get a really big jaw, you have to have really tiny arms. So you're sacrificing one thing, and that could be just one answer to why T-Rex had the little arms, because it was selecting for the super huge jaws, and it wasn't really using its front arms. Another example of this could be deer antlers, and there have been a couple deer recovered from they died around the time of the Ice Age, and they actually plotted them. They did a regression analysis on it in terms of antler size and body weight and survivability, and it turns out that the genes which control antler size also control body weight. Now, if you know anything about deer reproduction or anything, the antlers are very important, and individuals with large antlers are extremely desirable and much more likely to pass on their genes. Unfortunately, the gene that's controlling antler size also controls body size. So in order to get large antlers, which are desirable, you're also going to get a ridiculously large body. And if you've got a ridiculously large body, you're not going to be able to, it's going to be much harder to stay alive because you're going to be requiring much more food. So there's a certain limit. So you could ask, well, why wouldn't the antlers just grow to relative infinity? I mean, they'd be great. Females wouldn't stop mating with them. And the answer to that is because of pleiotropy. The body would get too big, and it would just be too much of an energy drain. So next time somebody asks you why a certain trait evolved in a certain way, be sure to keep these concepts in mind, as they're most likely the answer. Lastly, I had one quick question. If any of my listeners happen to know anything about servals, jungle cats, or savanna cats, please let me know because I'm really interested and I have a couple questions for you. Thanks again as always, guys. The seventh part in my How Evolution Works series is going to be discussing speciation, which is the natural process by which new species emerge. Over the past 100 or so years, there have been hundreds of documented, observed examples of new species emerging. In fact, it happens so frequently that there are even different types and categories of speciation to describe how this occurs, and I'll be going into each of those types as well as examples of them happening in this video. Before I begin, however, it's important to understand exactly what speciation is. Nobody is suggesting that a dog will give birth to a cat or anything like that. That is simply not how new species emerge. Instead, it's a gradual process, although it can be quicker in certain situations than others. For useful metaphor or analogy to exactly how this can happen, please check out the linked video. It's also useful to begin by defining exactly what a species is. Now, fortunately for biology, there's really not a lot of ambiguity since most biologists use what's called as the biological species concept. That is, two organisms are considered to be the same species if they can produce viable offspring. That is, if they can interbreed and produce offspring, then, well, they're the same species. If they cannot, then they are not. Now, note that this is not necessarily based on exactly how organisms look. One good example of this are the happy face spiders. They look completely different, but they can interbreed and are hence the exact same species. Overall, there are two major evolutionary processes by which new species can be formed. The first is simply general speciation, which I'll discuss in a minute. The second is called polyploidy, which results in a relatively instant creation of new species. Now, because this involves genetic abnormalities, it typically involves plants, and I'll be discussing that at the latter portion of the video. As far as general speciation is concerned, there are four types: allopatric, peripatric, parapatric, and sympatric. For the purposes of this discussion, however, peripatric and parapatric are so similar that we'll simply discuss peripatric. So to begin, allopatric speciation occurs when one large population is separated into two smaller populations by, for example, a barrier. Let's say that there's one large population of birds, and a mountain range pops up in the middle of their population, preventing the birds from flying and mixing with the other side. Over time, because the two populations aren't mixing genes, differences will accumulate and thus lead to new species. Perhaps the most famous example of allopatric speciation occurs in Darwin's finches. Now, when the Galapagos Islands formed, it provided a new habitat for the mainland finches to go on and diversify. Now, these new islands had many different habitats, and the finches could modify and select and fit into each different niche if they modified their beaks. So what do we see is we see the main finch type, but we see differing beaks allowing them to eat different foods. Some of them have large beaks for eating larger seeds, some of them have smaller beaks for eating smaller seeds. Some have beaks that are adapted for eating insects and nectar. Some have beaks like woodpeckers. And this modification is basically occurring through allopatric speciation. And no, they are not able to mate with each other. Another example of allopatric speciation occurs in house mice species on the island of Madeira, which is a Portuguese island off the northwest coast of Africa. These mice demonstrate allopatric speciation because they are physically separated by different valleys leading down to the sea from the mountainous mainland. Now, when one actually examines these mice, you'll find that there are 6 distinct populations, and each of them have a distinct karyotype, or number of chromosomes. These number of chromosome differences resulted from Robertsonian translocations, which are basically errors during reproduction where 2 chromosomes fuse. This is also the same thing that happened, by the way, in the Ken Miller of Apes and Humans video, which I've linked several times, where the process of the fusion is not necessarily creating the new species, it's merely an artifact of reproductive isolation. Now, this will also prevent the 2 populations from mixing, hence new species formed. So here you have a clear example of new species being formed. They've got different numbers of chromosomes, can't interbreed, and they came from the same common ancestor on the mountain on the island. The second type of speciation we'll be discussing today is called peripatric speciation. Now, with allopatric speciation, which occurred when, like, say, a mountain range was dividing one large population into 2 smaller ones, peripatric speciation involves one population and fringe members of it exploiting a new niche and accumulating enough differences to prevent breeding. So the key difference here would be that with allopatric, you have a physical barrier separating the two, whereas with peripatric speciation, there's a new niche. Let's say, for example, that a new type of nut or a new type of tree with a new type of nut that a certain bird was eating started to appear on the fringe of a population. Well, certain members of that population with larger beaks could be selected to eat those nuts. Over time, they could become specialized to only eat those nuts, and stop interbreeding with those who do not eat those nuts. Over time, enough genetic differences will accumulate, and boom, you have a new species. There are several documented examples of this happening as well. Another wonderful example of peripatric speciation occurs in the Australian bird Batroica multicolor. Now, I won't really get into the details of the speciation event, however, I did stumble upon a really neat paper while researching this video discussing the molecular phylogeny of it and tracing its ancestrage and lineage through mitochondrial DNA, and I'll link that right now. Another example of peripatric speciation can be found in the London underground mosquito, or Culex pipiens, which basically emerged over the last few decades when the London underground system was built. Basically, overground species of mosquito, or an overground species of mosquito, colonized, went underground, exploited the new niche, and it now is very different from its predecessor. The new species has completely different mating behaviors, it's therefore difficult to mate, and it also shows genetic drift. Here are sources citing that information. The last type of general speciation I'll be discussing is called sympatric speciation, which is where species diverge while they're inhabiting the same place at the same time. One example of sympatric speciation that's occurring actually right now exists in that of hawthorn flies. Now, the hawthorn flies, are also known as the apple maggot flies, are their normal hosts are the hawthorn trees. They're native to North America. But sometime in the early 1800s, they began to infest apple trees. Now, what happens when we take a look at these two different populations, that is, one population infecting hawthorns and the others that are infecting the apple trees? We find that there are significant genetic differences between them. For example, 16 out of the 13 Alzheimer's mellici, if you're interested in the specifics. Also, there are major differences in maturation. The species of apple flies tend to mature at 40 days, where the ones infecting the hawthorn trees mature at 54 to 60 days. So this is the middle, or the beginning rather, of a speciation event that we can actually sit back and take time and watch. It's occurring in nature. We've got species of these beetles, or examples, or specimens rather, of these beetles dating back hundreds of years, so we can see exactly what they look like before, and we can watch the changes unfold. Now, a really interesting example of sympatric speciation can be found in the three-spined stickleback, in which there are two species. One is a larger benthic species that has a large mouth and it feeds on prey, large prey rather. Now, there's also a smaller species which has a smaller mouth and, of course, feeds on plankton. Now, some interesting things about this are that the DNA analysis shows that each lake was independently colonized sometime after the last ice age by a marine ancestor. DNA analysis also shows that the two species in each lake are more closely related to each other than they are to any of the other species in the other lakes. But nevertheless, these two species are reproductively isolated and will not mate with each other. They are separate species. Now, something very interesting happens when we place them in aquariums, and that is that the larger species from one lake will spawn with the larger species of another lake, and the smaller plankton-eating species of another lake will spawn with the smaller plankton-eating species of another lake. But of course they will not mix. The last thing that I wanted to touch on here at the end of the video was discussing what factors favor the accumulation of genetic differences, because keep in mind, these accumulation of genetic differences between two populations is exactly what drives speciation. There are several things. First of all, a small population size. This greatly enhances speciation as well as genetic drift. A second thing is assortative mating, which occurs within a species, and it's basically the tendency for certain individuals to mate with individuals like themselves. For example, in a species of beetle, the larger individuals of that species will like to typically mate with larger females. Same thing with, smaller males will like to mate with smaller females, and this can greatly enhance and provides a unique opportunity for sympatric speciation. This can also be demonstrated experimentally in the case of flower beetles. In the laboratory, that is. There were researchers in 1981 took a population of flower beetles and divided them off of each generation. They would take the 8 largest males, the 8 largest females, and have them mate, and then they'd take the 8 smallest males and 8 smallest females and have them mate. And they would basically let this go on, the large mating with the large, and the females, and the small rather mating only with the small. And they continued this for 15 generations. At the end of it, it was demonstrated that assortative mating had occurred in half of the experimental lines. A researcher named Diane Dodd also repeated this experiment, only using Drosophila or fruit flies as her agent. What she did is she took one large population of fruit flies and arbitrarily divided them up, fed half of them only maltose food and the other half only starch food. And now these starch versus maltose subpopulations were kept separate and allowed to breed, and many generations were able to pass. What she found is when she put them back together, those raised on maltose food would not mate again with those eating only starch food, even though they came from the same population to begin with. So this is the formation at the beginning of a mating barrier, which would perfectly cause speciation because, again, it's preventing genes from flowing back and forth. Lastly, a logical question arises, and that is, if these two populations are separate, what's to prevent them from coming back together? And the answer is isolation mechanisms. An example of this would be birds having complex dances. If the other bird is a member of a different species, it won't know the dance and it won't mate with it. This is also seen in Galapagos finches, who select their mates based on different beak sizes. Different species have different sized beaks, and thus they won't mate. Another thing is that this is often seen in insects, is that the sex organs physically and structurally are not compatible. Another thing that could happen is that the sperm could fail to reach the egg. These are termed prezygotic and postzygotic mechanisms, or isolation mechanisms, and it's actually a fairly good read if you have the time. So to refresh from last time, there are several steps in the process of the formation of a new species. Assortative mating develops, which is where large organisms will mate with large organisms, small organisms will mate with smaller ones, and this is within a species. It's basically the tendency for like organisms to mate with other like organisms. After that, we see isolation, which we can see in the development of mating barriers, such as mating dances, hybrid sterility, things like that, which basically prevent different types of organisms from mating. After that, we can see differences accumulating. And one thing I mentioned previously, that smaller populations are subject to more, or speciate quicker than large populations, and the reason for that is that small populations are subject to more genetic drift. And if you want to think about it from a statistical point of view, the smaller the sample size, the greater the chance you have to get differences from the mean. Well, here, those differences from the mean can be thought of as genetic differences, and that's exactly why in smaller populations, genetical differences accumulate quicker. So after all that happens, we have a new species. That being said, it's not always 100% crystal clear cut exactly when a new species is formed. As I mentioned before, there are several steps, and organisms can lie at different positions on that gradient. One such example demonstrating this is a phenomenon known as ring species, whereby you can have, let's say that there are 8 different populations of a single species of an organism. This happens a lot in birds and parrots. There's one species in Africa, I forget the name though, but it's a type of parrot that this is commonly seen on. And each population will be able to mate with the populations directly adjacent to it. But, and this goes and continues all the way around, every population can mate with the one next to it. And populations at either end or across from each other are unable to mate. So this is one area which can be sort of gray in the question of, has a new species formed or not. The answer to this I think most biologists would agree that this is a species in the process of speciating and branching off. I'd like to shift the discussion now towards speciation in plants. For most plants, copper is relatively toxic, even in low amounts. It's not surprising then that copper-resistant plants would have an evolutionary advantage in areas of high copper. And we see that exactly around copper mines out in California. The yellow monkey flower, or Mumulus, for example, has recently been observed to have speciated into copper-tolerant types. Specifically, Mumulus cuprophilus is one of the examples. Basically, there were some mines that were last used around 1859, or were opened in 1859, and there are several new species of cuprophilus that are only found in copper-rich areas around those mines. They are new species and not wild type because when they are mixed with wild populations, the offspring and hybrids are inviable, so therefore they are a new species. Also continuing the discussion on plants, if you recall from the first video, I spoke of several different types of speciation events and how they can occur. The first part was general speciation, where I discussed sympatry, allopatry, things like that. But the second type that I didn't mention in the first video was polyploidy, which is an interesting phenomenon that's often observed in plants and continually gives rise to new plant species all the time, and animal species too occasionally, but it's much more common in plants. Now polyploidy results in an abnormal number of chromosomes. Often you have an organism will be 6N or 8N or even 4N, things like that. And if you recall, sperm and eggs are 1N, which means you have 1 times the number of chromosomes, and our body cells or our somatic cells are 2N, which means that the sperm and the egg fuse, each of them are N, so you each get 1 chromosome, fuse it, and you get 2N, which gives you the full complement. Well, most plants that we see around today are 4N, 6N, or 8N, and the question then arises, well, how can this happen? Again, for an overview of exactly how polyploidy works, take a look at this image right here. So the body or somatic cell typically has 2N, which is 3 pairs of chromosomes for this particular organism. Now, the gametes, egg or sperm, which are seen to the right of that in the middle, should just be 1N. That is to say that instead of having 3 pairs of chromosomes, which they do, they should just have 3 individual unpaired chromosomes. But what happens is if an egg that has undergone non-disjunction and a sperm that have undergone non-disjunction join, both of which are 2N, the result is 4N. And believe it or not, this 4N individual can occasionally be sterile. Typically, the infertility results when an uneven number of chromosomes are presented to an organism or contained in an organism because they simply can't match up. During meiosis, they have to pair up. You find these unevenly numbered organisms unable to pair because, again, there's not an even number, so some of the chromosomes are unable to pair. They don't have a match. But what happens in a case like this is because each of them has been doubled, although it's relatively rare, the organism is able to reproduce, and there have been many new species of plants produced this way. In fact, most crops are the result of polyploidy. For example, of some tetraploid crops that are 4 times, or not even crops, but salmon fish are tetraploids. For hexaploids that are 6 times, that is wheat and kiwi fruit. Octoploids are commonly seen in sturgeon fish. Decaploids, which are 10 times, are seen in strawberries, and the list goes on and on. So the moral of the story is that this is fairly common, and it is an easy way to produce new species that are viable. So, now on to observed instances of plant speciating due to this mechanism of polyploidy. The first would be in 1905, where Dutch botanist Hugo de Vries noticed an odd-looking plant in his collection of evening primrose. Upon further analysis, this odd variant had the chromosome number of 2N equals 28, which is twice the number of chromosomes that the typical wild type of evening primrose has, which is 2N equals 14. So we tried to breed this new specimen with the wild type and was found that they simply weren't able to be bred. As such, this new species was named Enothera gigas, and the wild type remained its original name of Enothera lamarckiana. Now for a fantastic example of creationist dishonesty. I typically go and look up rebuttals to my own arguments just to see, if they have any merit, if I want to see the other side's point of view to see if my arguments truly are accurate. I don't publish them or I don't produce them in my video if I don't find them to be that case. So I was looking at the Creationist Ministries International website for the rebuttal to this particular thing, and it was actually extremely interesting, and it's a perfect case of creationist dishonesty. I quote from the site, "The idea that these plants constituted an example of speciation is wrong, and it was realized at least as long ago as 1943, more than 6 decades ago." And they list a source there, number 7. Well, when you actually scroll down to the sources at the bottom of the page, it is talking about a cross between 2 completely different species of plants and how the offspring won't survive, but it's just in the same genus. That is the only thing. They're not even talking about remotely the same species. It's a paper from 1943 that they're referencing, and it is a failed cross between 2 completely different species. So what they're trying to do is take that paper and somehow connect it to this species. It's dishonesty at its finest. I mean, they're just counting on you not to take a look at the reference or not even to bother to read the paper. It's truly sad. Another example of observed speciation can be seen in the Tragopogon or goat's beard plants. In the early 1900s, 3 different species were transplanted to North America. They were the Parafolius, Dubius, and Pratensis. Now, in 1950, that's 50 years later, it was observed that 2 new species had emerged from those via hybridization. Myrrhis is a hybrid between Parafolius and Dubius, whereas Macellus is a hybrid between Dubius and Pratensis. Now, these only evolved once they got to North America and within 50 years. They're not compatible with either parent, and they are able to interbreed among themselves. So they are a brand new species, able to again, produce viable offspring. What's also pretty interesting is that it wasn't until the 80s, but it was observed that Miris had evolved independently at least 3 times, and this was confirmed using chloroplast DNA. The same thing is also true of Macellus having independently evolved several times. Furthermore, in the 1920s, a stable hybrid was actually formed between a radish and a cabbage. Now, these two organisms aren't even in the same species, but despite that, they were able to hybridize and make a sterile F1 offspring. So what happened to this F1 offspring is, well, they're sterile, but via non-disjunction, or the production of unreduced gametes, they were able to get offspring that were able to produce seeds, and thus created a new species. But unfortunately, the new plant had the foliage of the radish and the root of the cabbage, which is the exact opposite of what you would want. But nevertheless, a new species was produced. It was fertile offspring and unable to reproduce with either parent.
Keep in mind, we have the original population of lizards as well back in Italy. So suffice it to say, we actually know a lot about this. It's happening continually, and it's an ongoing process that we learn more and more about every day and every year. This video has been a pretty basic introduction into exactly what evolution is. Now, my next videos will be focusing more on the biology of evolution in general, and specifically the next one will be focusing on the 4 forces which drive evolution. So stay tuned, guys, and once again, thank you very much for your time. As I stated before in my prior video, there are 4 main forces which drive evolution. They are natural selection, mutation, genetic drift, and gene flow. Now, I'll go into all 4 of those at length. So to begin with, we have mutation. Mutations are random changes in an organism's DNA. Now, mutations can occur in several places. For example, it can occur in a somatic cell, which is a cell of your body. For example, you can have one of the skin cells on your arm undergo a mutation. That'll be influenced by you, and it won't have any effect whatsoever on your offspring. However, it can also occur in some of your gametic cells, such as your egg or your sperm. If you have a mutation in a gamete, and that egg or that sperm goes on to produce a child, that mutation will go on and be present in all of your children's body cells. Mutations can typically have one of 3 effects. Some of them are bad. They decrease the overall fitness or survivability of the organism and are not selected for. Now, the second type, the vast majority of them are neutral, meaning that they provide no change whatsoever to the organism's fitness, and it is neither helped nor harmed by the mutation. The third type, which is the rarest, are beneficial mutations. These help the organism by increasing its fitness and the likelihood that it'll reproduce and pass on that mutation. Now, specifically, there are several types of mutations in the sense of inversions, deletions, additions, insertions, and things like that. However, I'm going to be discussing that in my next video, which will essentially also give you guys a brief introduction of DNA. But until you guys understand what DNA is and how it works, me going into the nitty-gritty specifics of mutations really won't help you. So I'll leave it as is at this point. Nevertheless, mutations are random. A very important thing that people don't seem to understand is that an organism does not know it has to mutate. For example, let's say that there are a group of green beetles and there are some mutated brown beetles. The green beetles are the natural wild type, but due to a change in a pigment gene, it'll also produce brown. Now, let's say that there's a bird that can only see and is only used to eating the green beetles. It won't touch the brown beetles. Now, did the green beetle, did it know that it had to mutate and become brown so that the bird won't eat it? No, what happened is that it passively mutated and the environment selected for those which were brown. And this series of videos is designed to teach people exactly how and why evolution works. While I'll be offering plenty of evidence for evolution, the goal of this series won't exactly be to convince people of evolution. This is because evolution is open to about as much reasonable doubt as whether or not the Earth goes around the Sun, and time is better spent educating people on the specifics of it. As far as the level of technicality that these videos will be discussing, they're pretty much suitable for everyone. I'll be discussing everything from the intro to DNA, intro to genetics, all the way up to epistatic interactions. So whether or not you're a biologist, or you have absolutely no training whatsoever in biology, you'll still have a lot to learn, and hopefully the information will be presented in such a way that anyone can understand it and find it useful. I've been doing this long enough to realize that there are 3 types of people involved in the public debate. The first are people who are open-minded, intelligent, and understand the evidence for evolution and subsequently accept it. The second type of people are those who are open-minded, but they are not really sure of the evidence and they don't know what to think. They're fence-sitters, so to speak. However, if they understood the evidence, they would accept evolution, and they do have open minds. Thirdly, there are those types of people who sadly there is absolutely nothing whatsoever that you could ever show them. They will not accept evolution. Their minds are about as open and porous as a sheet of glass. There's really nothing whatsoever that you could do. As Ken Miller often points out, his colleagues will often come up to him and say, well, what if we just showed them this experiment, or what if we did this? Would they then accept evolution? And the answer is simply no. When these people view evolution as the way they do, and they're not willing to accept or process any data whatsoever, there's absolutely nothing that you can do to get through them. And as such, I'm assuming this attitude, and I'll be pursuing it and not addressing these people in my video. Quite simply, you can't fix stupid. This video, therefore, is aimed at the other remaining two people, those undecided and those decided. Now, those people who are undecided will get educated and be able to see the evidence so that they'll be able to better make an informed decision. Now, those who already have decided but don't really have extensive biological training will get that, for free nonetheless. So, to begin, this first video is essentially going to be discussing exactly what evolution is, and more importantly, what it is not. The National Academy of Sciences recently called evolution the unifying theory of biology. This is because it unites virtually every independent discipline in biology so that scientific advances made in one can be universally applied to all others. Now, this has led to an explosion of research, and which is why evolution truly is at the core of biology. Famous biologist once said that absolutely no guys. Also, another link that I would highly recommend you guys is this Talk Origins FAQ. This essentially rebuts virtually any creationist argument that you could ever dream of. If there's something so obscure that they're very good at pointing out randomly, you can find it here typically. And it's a wonderful resource, and highly encourage you guys to take a look at it whenever you run into any issues whatsoever if you don't have much scientific training, or if you're simply interested in browsing. Your time will be well spent. In a biological sense, evolution is best described as descent with modification. This leads to changes in gene frequency within a population over time. The idea is that an environment selects for an organism that's best adapted for survival in it, and they're encouraged to breed. This concept leads to a change in gene frequency over time as well-adapted organisms breed at an accelerated rate, and their genes constitute a larger portion of the gene pool. As time progresses, populations become isolated, subdivided, and undergo different changes at different rates as they become very well suited to their environments. Now, after two populations will accrue enough differences, they'll no longer mate. When this happens, a new species is formed and they're said to have speciated. Now, this happens more common and rapidly than most people think. For example, scientists have observed 40 speciation events in the last 15 years alone. This is directly influenced by many factors, including generation time, which I'll get into in later videos. However, the point being that it's very common. Actually, some interesting news about just this topic came out last week. In 1971, 5 adult pairs of Italian wall lizards were transplanted to the island of Croatia. Now, they were an invasive species and essentially overran the indigenous population there. Go scientists. Well, the Croatian War of Independence broke out in 1971, or shortly thereafter rather, and nobody was allowed on the island, at least not scientists, to study the effects until recently in 2004. What they discovered was that not only had the new lizards overrun the island, but they had completely evolved. And not only had they evolved, but they had evolved at a rate that even surpasses what evolution would predict. In other words, this is a phenomenal example of lizards evolving at a rate that's much, much higher than one would predict. In fact, these lizards the lizards that were found developed cecal valves, which are muscles between the large and the small intestine that slowed down food digestion in fermenting chambers, which allowed their bodies to process the vegetation cellulose into volatile fatty acids. So essentially, they evolved an expanded gut to allow them to process these leaves. This is adding a brand new structure in just 30 years. This is a wonderful example of a new species forming and the addition of a brand new structure, which is not originally present in the organism's genome, because nothing in biology makes sense except for in the light of evolution, and this is 100% correct. So with that being said, I'll furthermore dive into exactly what evolution is and is not. To begin with, evolution does not state that human beings came from monkeys. It states that we share a common ancestor which is completely different. Second of all, evolution and God are perfectly compatible. Evolution says absolutely nothing about the existence of a deity whatsoever, and as this recent survey shows, 70% of scientists believe in a God. Furthermore, 99.98% of biologists accept evolution. So as you can see, there's clearly no discrepancy whatsoever. In fact, the past several popes have accepted evolution, and it's only in conflict with a literal interpretation of the Bible. Thirdly, evolution doesn't deal with how life arose on Earth. It only deals with life once it's already here. The same holds true for the Big Bang, many other theories which are commonly confused with evolution. The Big Bang and abiogenesis are theoretical physics and chemistry respectively. They have absolutely nothing to do whatsoever with biology and are about as related to evolution as they are the cell theory. Evolution is not something that one believes in, it's something that they accept. This is because evolution is a scientific fact. You don't believe in gravity, you don't believe in cells, you accept it and you acknowledge it. Now, evolution is not a belief also because it's testable, it's falsifiable, and it makes predictions. Hence, it requires absolutely no faith whatsoever, only acknowledgement. Next, evolution is not contradicted by really any evidence. Virtually every single ounce of evidence or observations that we can make about our natural world support evolution. Evolution was also not contrived to undermine any religion whatsoever. Basically, our awareness of it grew as we tried to make sense of the many observations of life that happened in a testable way. Evolution is not an accident or random process. There are some random components of it, however the vast majority of it is due to natural selection, which is by definition a non-random process. Evolution is also not something which happens to specific individuals. Instead, it happens to populations. Evolution is not something that only happened in the past. It's a current process, it has happened, it's happening now, and it will happen in the future. A quick comment before I progress too much. If in this video you find something that you want to learn more about, don't hesitate whatsoever to make a comment. I'm pretty good about responding. Furthermore, you should also take a look at the berkeley.edu website. They've got a fantastic evolution page, and it'll explain and reinforce a lot more of the concepts that I'm explaining to you. Made those which were brown more likely to pass on their brown genes. The second force driving evolution is natural selection. Natural selection is, by definition, a non-random process. Another common mistake is that people seem to think that the organism does the selecting, or it's selected for one way or another, but it is actually the environment which is doing the selecting. As I said before, mutations provide the raw material, the raw genetic material for natural selection to work on. You can kind of think of it as mutations creating a static image and natural selection being the refinement of that image to produce a picture. Actually, take a look at this image that I'm showing right now. It's a computer model made by CDK007. I highly suggest you guys check out his vids, which start off by with a random static field, and by selecting only the ones which are likely to improve and closely match the picture that's desired, we end up with an overall South Park picture, or a picture of a South Park character as the net result. Although I absolutely loathe using the term survival of the fittest, this is essentially what it's referring to. In a nutshell, an organism which has genes which allow it to be very well adapted to its environment is more likely to be able to survive and pass on those genes. Over time, those genes will represent a relatively larger percentage of the gene pool of that population. The third force driving evolution is genetic drift, which is a random process. In all actuality, it probably is understated in the sense of genetic drift probably has a bigger effect on population genetics than most people realize, but nevertheless, it is a random process, and it's often overlooked, but it's very important. Now, evolution works best in very, very small populations, because in those populations, there's a lot less averaging, and big changes can accrue very, very quickly. Kind of think about it with a law of averages, and if you've ever done any statistical work, think of outliers. For example, let me say that I'm taking the average weight of a baby, and I have 5 babies. 4 of them weigh 15 pounds, and 1 of them weighs 3 pounds. Now, this is completely hypothetical, but think about the effect of that 3-pound baby on the average weight of all those 5 babies. Now, on the other hand, let's say that I take the birth weight of 100 babies, and 99 of them weigh the 15 pounds, and there's still that 1 3-pounder. So if you have the large sample size of 100, the average birth weight will be much closer to the reality of 15 than it will in that 5, or in the group of 5 individuals. Now, the 3-pound baby is termed an outlier because it greatly